Good day, this is the Professor, CBMD PhD, and today is Saturday, August 12th, 2023. It is 6:21 and 14 seconds, AM Eastern Standard Time, and today's research, the liver. So let's jump to it here. Humanized liver in mice reveals roots of chronic diseases. Date August 9th, 2023. Source, Yale University. Researchers have created a functional humanized liver in living mice that will help scientists find human-specific mechanisms for regulating cholesterol levels and potentially for treating chronic liver diseases afflicting tens of millions of people in the United States. As chronic liver diseases such as alcoholic and non-alcoholic liver disease, cancer, viral hepatitis, fibrosis, and cancer affected more than 1.5 billion people worldwide. In the U.S., an estimated 30 to 40% of the population has been diagnosed with non-alcoholic fatty liver disease alone. Yet liver disease has been difficult to study in animal models. The livers of mice, for instance, perform different functions than those of humans. Inside the liver, multiple human cell types talk in their own language. Mouse and human cells talk in different languages, but we have enabled human liver cells to speak in their own language within living mice. The scientific investigators have microbiologists use progenitor cells and mature cells known as hepatocytes from a human liver to create a complete human liver in a mouse model. The humanized liver, researchers said, developed in a similar size, adjusted shape, and carried out similar cellular functions as a healthy human liver. The cellular functions in a humanized liver can also be manipulated to mimic human fibrosis and non-alcoholic fatty liver disease. The researchers also found that essential liver metabolism is controlled by activity in endothelial cells which line blood vessels that feed the liver. Those endothelial cells, they said, secrete a signaling molecule called WNT, which regulates cholesterol transport to hepatocytes for the synthesis of bile acid. The transport of cholesterol to hepatocytes is an important mechanism that reduces excess blood cholesterol levels in humans. According to researchers, the humanized liver model can be used immediately by drug companies seeking to assess safety of experimental drugs designed to treat chronic diseases. However, a long-term goal is to find ways to predict, prevent, and treat all liver diseases, which take such a huge toll on individuals. And this is the Professor, Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.